Hello everyone, my name is Sarah Gordon and I will be discussing with you today bone proteomics and forensics, and specifically how we can look at the bone proteome to help determine the postmortem interval of submerged remains. And proteomics is just a branch of molecular biology that studies the set of proteins expressed by the genome of an organism. So it's easy to think of the proteome as being like the genome because the genome is just the complete set of all the genes within an organism. So the proteome is just the complete set of all the proteins within an organism. And so when we say bone proteome, that just means we're talking about all of the proteins that are expressed in bones specifically. And first, before we get started, I'm going to give you some definitions and explanations of some key terms, like what is postmortem interval and why is it so important, and why are we singling out the bone proteome? And then I'm going to go over a few of the study's hypotheses, because the proteome is massive and there's a lot of different things that are going on in this study. And then I'm going to go over the study's approach and follow it up with our results. And lastly, I'm going to give you some take-home points or the findings in this study that I feel are the most important. All right, and so to get started, what is PMI? PMI is the postmortem interval, which is the estimation of time elapsed since death. And PMI is crucial in criminal investigations because it allows the investigators to estimate when the victim has died, and so they can narrow down suspects accordingly. However, PMI is extremely hard to determine because there are so many variables that can influence a person's rate of decomposition, like the type of soil, the temperature, and the presence of scavengers and insects. And we currently have a few methods in determining PMI, like observing the presence of rigor mortis and by looking at the insect succession within a body. And rigor mortis is the stiffening of the limbs post-mortem. And so observing rigor mortis can help us determine PMI because it usually sets in within around 2 to 6 hours post-mortem, and it lasts for around 12 hours. But 2 to 6 hours is still a pretty big window of time, and there are so many things that can accelerate the onset of rigor mortis, so you can't look at it alone. And forensic scientists often use ADD, or accumulated degree days, to help determine PMI. And so we know that insects are temperature dependent, so that means that just the warmer it is, the faster they grow, but they also have a threshold temperature. And so that means that if the temperature dips below this threshold temperature, the insects will hibernate and not develop at all. So if an insect has a threshold temperature of 40 degrees and the temperature outside is 32 degrees, the insect isn't going to grow. And so ADD is a way to measure the passage of time and temperature simultaneously by recording the average temperature and using it to determine the developmental stage of insects within a body. And I've also included a little diagram about insect succession at the bottom here. And so insect succession just refers to the stage of development an insect is in and how many generations are present in the body. And so these methods are extremely examiner dependent, and a lot of them can't be applied to bodies in advanced stages of decomposition. But recently we've seen more success in using molecular-based approaches involving the analysis of proteins and RNA to characterize the decomposition of bodies, specifically with advances made in mass spectrometry technology. So proteomics has been increasingly used with more success in determining PMI. Okay, and so that previous slide was all about determining PMI in bodies that were found in terrestrial environments, so like in soil. But how do we determine PMI in bodies that are not found in terrestrial environments, like underwater? And so we use the term PMSI, or postmortem submerged interval, when talking about bodies that are found in water. And like with PMI, PMSI is also extremely difficult to determine because there are even more variables to consider. So like tide, salinity, water depth, and the bacterial and chemical composition of the water. And we use some of the same methods as we do with PMI, like calculating the accumulated degree days. And we also use something called the aquatic decomposition score, or ADS. And ADS is most commonly used to estimate the PMSI of bodies found in saltwater or freshwater. And they do this by measuring the degree of decomposition in several areas of the body. so like the face, neck, and limbs. However, these approaches are also very limited because with bodies that are submerged underwater, it's often that you don't recover all of the parts of the body, so you can't use this ADS scoring method. But what we do know is that as long as any piece of the body is recovered, proteins can be recovered as well. And because we know that proteins degrade as postmortem interval or time increases in terrestrial environments, we can potentially look for any degradation patterns in these proteins that may indicate how long a body has been submerged. And to overcome a lot of the previously mentioned limitations, bones can be a really good biomolecular target because they survive the longest post-mortem and they're pretty stable against any external forces. And so proteins especially survive longer within the bone structure post-mortem. And so bones are made of two phases, an inorganic phase and an organic phase. And the organic phase contains mainly proteins. And so within this organic phase, there are two types of proteins, collagenous proteins and non-collagenous proteins. And non-collagenous proteins are tightly attached to the mineral matrix of the bone, which makes them pretty stable and well preserved. So they are really good candidates for forensic studies. And we already know from previous studies that non-collagenous proteins show a pattern of degradation as post-mortem interval increases in terrestrial environments. And specifically, FET2A has been found to be a good biomarker for determining the age of a body. And this is because as time increases, the abundance stays constant, and individuals that are around the same age show similar abundances of FET2A. So you can look at the abundance alone of FET2A and determine the age of a subject. So this is not the same as PMI because PMI is just the time elapsed since death, but FET2A can tell you exactly how old the subject is. And we also know that deamidation ratios of biglycan, a small protein found in bones, increases as postmortem interval increases. And so deamidation is a chemical reaction where the amide side chain of asparagine and glutamine is removed or converted to another functional group. But what we need to know about deamidation is that it is one of the most common degradation reactions of proteins. And so if the deamidation ratio is high, that means that the proteins are degrading. And from that, we may be able to look at a deamidation ratio of a protein and pinpoint exactly how much time has gone by since death. And so from all of that, we can narrow down what this study is looking at. And so first, we're going to see if the type of water can influence the proteome. So will there be different abundances of certain proteins in saltwater or freshwater, for example? And second, we're going to see if PMSI influences the proteome. So as time increases, will protein abundance decrease? And then depending on whether a PMSI affects the proteome, what are some good biomarkers that can help us determine PMSI? And lastly, we're going to see if FET2A is a good biomarker for determining the age of a submerged body, like it is in bodies found in terrestrial environments. Okay, and now we can start getting into our approach. And overall, there are two conditions in this study. So PMSI, 1 week or 3 weeks, and type of water. So you have pond water, tap water, salt water, and chlorinated water. And so 22 frozen mice carcasses were used for this study, and they were all defrosted and then put in their different conditions. And so let me get it. Okay, and so you have 16 were submerged in these different water and time conditions. So for example, you would have 2 tap water 1 week and then 2 tap water 3 weeks. And so you have that for each condition. And the tail of each mouse was taped down at the same level within each water bottle. So each mouse was at the same water depth. Okay, and so with our 6 non-submerged mice, 2 were allowed to defrost and then immediately after were analyzed. So this is our 0-week control. And the remaining 4 mice were set aside in clear foil boxes for the same time conditions of 1 week and 3 weeks. And these 4 were never submerged in water, and they were each put in the foil box at the same temperature and kept for however long. And then after the specific time had passed for each condition, the mice were removed and then their tibia were retrieved. And in this study, they had 22 tibiae. Okay, and so here's a diagram that lays out each step in this study. And so this top part up here is just our independent variables. So you just have the time, the 0 week, 1 week, 3 week, and then the type of water. And so then after the tibia were collected, the bones were decalcified, and then the proteins were extracted and digested into peptides. And then they used liquid chromatography tandem mass spectrometry to analyze these peptides. And then the peptide mass spectra that were produced were put in a search engine, which identified the proteins and assessed their relative abundances. And then at the bottom here, we just have our dependent variables. So the effective PMSI, water type, and deamidation. Okay, and so this is a principal component analysis plot. And these are used when you have very large data sets. And so the PCA plot reduces a large set of variables into smaller sets that still contain most of the data. And normally on a graph, you have a 2-dimensional plane. So an X and Y axis. But in cases like with this data, you could potentially have 30 dimensions for your 30 different variables, and it's impossible to comprehend a graph like that. So a PCA plot combines variables that you can have on a 2-dimensional plane. And PCA plots are really good at showing similarity, but in order to get this 2-dimensional easier to look at graph, you'd have to cut some of the data out that doesn't contribute to the overall diversity in the data. So that's what these numbers on our X and Y axis mean. And so if you add these 2 numbers together, so 21 and 52, they're a little bit more than 70%. And so that just means 70% of the original data is being displayed here. And so that's all the logistical stuff behind this graph. But all we need to do is just kind of ignore all of that and just visually look at it. And so each of these different colored clusters represent our different PMSIs. So this blue here is our 0-week control where the mice were only defrosted. And then this orangey red color is our 1 week. And then this yellow color here is our 3 week. And clusters that are closer together are more similar. And so by just looking at the position of all 3 of these clusters, you can see that the red and yellow are a little closer together than the red, yellow versus the blue down here. So this just means that there's a big difference between our submerged samples, the 1 and 3 weeks, and then the 0 week. And additionally, these 2 up here are a little farther apart from each other. So they're not right on top of each other. So they're still a little, they're similar, but not so similar. And so that just means that there is a slight difference between the different times. And so the bone proteome of the mice submerged in water is modified differently based on the length of time they're being submerged. Okay, and so we're back to much more simpler graphs. And there's a lot of figures in this study, so I have this little marker at the top that you can keep track of where we are and what we're talking about. So right now we're talking about postmortem submerged intervals still. And aldoa is an enzyme involved in glycolysis, and it is explicitly expressed in skeletal muscles. And so this figure shows the relative abundance of aldoa in each condition, with the control being our 0-week defrosted mice, then each water type at each time. And the water types are abbreviated, with FM being tap water, SM being salt water, PM being pond water, and CM being chlorinated water. And the abundance of aldoa significantly decreased between 1 week of salt water and 3 weeks of salt water. And so from this, aldoa may be a useful biomarker in determining PMSI because of its close proximity to the bone, and its continuous degradation over time. And however, further studies should still be conducted to determine how long this protein can be recovered from skeletal muscles in submerged bodies. And like the previous figure, this one displays the relative abundance of FET2NA. And FET2NA is a plasma protein, and as I said earlier, FET2NA has been found to be well preserved and can indicate a sample's age. However, FET2NA can be used to determine a sample's age because its abundance stays relatively constant post-mortem. And as we can see from this figure, it decreases at 1 week and then it increases at 3 weeks in all the different water types. It is also more abundant in the control samples, and this is probably because of the presence of peripheral blood, and it would be diluted in the water of the submerged samples. And all of the mice were about the same age, so because the abundances did not stay constant at each time interval, FET2NA is most likely not a good biomarker for chronological age, as was previously found in terrestrial environments. And so this figure is also very similar to the previous two because we're still just looking at relative abundance on the y-axis and the water types and time intervals on the x-axis. And this time we're looking at peptidyl cis-trans isomerase A, or PPIA, and it is an ubiquitous protein that accelerates other proteins folding. And PPIA is significantly more abundant in this 0-week control than in weeks 1 and 3. And there was no significant difference between the 1 week and 3 week in any of the water types. And it has never been previously reported in studies about body decomposition, but it shows a quick degradation in aquatic environments. So because of this, PPIA could be a good biomarker in determining whether a body was submerged or not. And so here we have another PCA plot, and it is nearly the same as the first one, except the controls were removed because we already know that there is a difference between the submerged and non-submerged samples. And so because we know that there's a difference, we can look at specifically if there's a difference between the types of water. And so like before, we're just going to look at where the clusters are and just ignore the axis stuff for now. And so these clusters right here are all on top of each other, so that means they are very similar. And so the proteome changes in pond water, chlorinated water, and tap water are very similar. And so the saltwater cluster up here is farther apart than the other three. So it appears that there is a slight difference between them because it's farther away. However, so now we're going to start talking about the x and y axis again. And the x-axis explains about the x-axis explains about 15% of the data, and then the y-axis over here explains about 37%. So if you add that together, it's still less than 50%. So this just means that only 50% of the data is being explained, because if you remember, as I said earlier, a PCA plot tries to cut down how many variables are being shown so that they can easily put it on a two-dimensional graph. And so with these conditions, there's only 50% of the data being represented. So this just means that more of the difference between the clusters is explained by time and not by water type. Because if you remember, the first graph had a 70% representation between the axes. So more of the differences between the clusters is explained by time and not water type. And so, yes, there's a slight difference between saltwater and the other types, but not enough of a difference for it to be a really big deal. Okay, and so now we're going to be looking at deamidation ratio. And just to recap, deamidation is one of the most common degradation reactions of proteins. So if the deamidation ratio is high, that means the proteins are degrading. And so on this figure, we have a FET2NA peptide on the left and an IGF-BP5 peptide on the right. And IGF-BP5 is an insulin-like growth factor binding protein. And so on the y-axis, we have deamidation ratio. And we're looking at all the different times, so 0, 1 week, and 3 weeks. And in FET2A, we're looking at salt water, and IGF-BP5, we're looking specifically at chlorinated water. And there were no significant differences in these peptides in any of the other water conditions, so that's why salt water and chlorinated water are being singled out. And so in both, we can see that there is a significant increase in deamidation ratio between 1 week and 3 weeks in the FET2A peptide. And then in the IGF-BP5 peptide, there is a significant increase between 1 week and 3 weeks in the chlorinated water. And so these two peptides could be biomarkers in these specific water types. Okay, so this figure is like the last one. So we're still looking at deamidation ratio on the y-axis. And this time we have a different FET2A peptide and a polypoprotein E, or APO, on the right. And unlike the other figure, we're looking at each water type, but specifically at 3 weeks postmortem. And in the FET2A peptide, you can see that there is a significant difference between salt water and pond water, and then another significant decrease between pond water and chlorinated water. And so APO also has a significant difference between tap water and then pond water. And if you notice, they're both in pond water. The difference is between pond water. And it's likely that the deamidation ratios in these two peptides was higher in pond water because of the bacteria that is usually present in pond water. However, identifying the bacteria that is usually present in pond water was way beyond the scope of this study. So the means to confirm body submersion in pond water is very limited at this stage. So more research is specifically needed on pond water to clarify the effects of pond bacteria on the deamidation of bone proteins. Okay, so in this last figure, we're looking at the deamidation ratio again on the y-axis, and on the x-axis, we have the coagulation factor 7 peptide at each time interval in specifically tap water. And so if you look on the left graph, we have there's a significant increase between the defrosted mice 0 week and the 1 week time condition. And so if you look on the figure on the right now, this is comparing specifically the terrestrial control. So those 4 mice that were kept in the like that foil box for 1 week and 3 weeks versus all of the different water mice at 1 week and 3 weeks as well. And so if you look, you can see that there were no deamidation reactions in the non-submerged samples, but there were some in the submerged samples. So this just means that the presence of deamidation of the F7 peptide can indicate that a body has been submerged in water. And so to wrap up, here are some take-home points. Firstly, we know that the bone proteome changes are different in submerged remains than in non-submerged remains. So, so being submerged in water does affect the proteome. Second, we know that the postmortem submerged interval affects the bone proteome more than the water type the subject is submerged in. So how long a body is underwater affects the proteome more than if it was tap water or pond water, for example. And we also found a couple of potential biomarkers. So we can look at the abundance of aldoa and the deamidation ratio of FET2A and IGF-BP5 to help us determine PMSI. And if we notice there are deamidation reactions in PPIA and F7, we know that a body has been submerged in water. So it doesn't help us necessarily with determining how long it's been, but we just know that a body was submerged, period. And lastly, more research is needed on all of the above points. And this is because this study was only conducted over a 3-week period, so that's not really a long period of time. So we need to see how long we can observe these proteins in submerged remains, as well as studies comparing the same type of water from different geographical locations. And lastly, I just want to say thank you for your time and thank you for watching.